In this particular video, we will be going over what the Hardy-Weinberg equation is. We will not be attempting to solve any Hardy-Weinberg problems. I do have a different video where I show you how to use the Hardy-Weinberg equation in various scenarios. So Hardy-Weinberg was developed independently by an English mathematician and a German physician. They used algebra to explain how alleles, gene frequencies, predicted genotype and phenotype frequencies in a population. Hardy-Weinberg many times is used in evolution and it is used in genetics. Understand that Hardy-Weinberg is for predictions. It deals with probable outcomes. And so with genetic ratios and dealing with the likelihood of individuals inheriting certain conditions, this is a very good equation to look at. When dealing with evolution, this does not deal with macroevolution or the conversion of one species to another species. What it deals with is probability of adaptation of species within certain environmental conditions. So this deals with adaptation or microevolution, and it deals with genetics and how individuals inherit certain conditions. The Hardy-Weinberg equation is mathematically written right here, where we have p squared plus 2pq plus q squared equals 1. Now the p is the allele of one, it's the frequency of one allele or one gene. The q is the frequency of the other gene or the other allele. The p typically is for the dominant and the q is for the recessive. The mathematical equations that we understand in order to solve these problems is p plus q equals 1. Then we have p squared, q squared, and 2pq. Now the 2pqu, this is for the carrier state. The p squared is for the homozygous dominant. The q squared is for the homozygous recessive. And again, what this is showing you here is that the p squared is going to be for the homozygous dominant. The q squared is going to be for the homozygous recessive, and the 2pq is going to be for that carrier state. Here we have a problem that you can look at solving, where you can have normal fingers as big D, big D. You could have normal fingers as big D, little d, or you could have short fingers as little d, little d. The p frequency is 0.7. The q frequency is 0.3. And so we could have the p squared, 0.7 squared is 0.49. That is the likelihood, 49% of the 49% chance of having big D, big D. The 2pq is 2 times 0.7 times 0.3. Multiply that out, 42% likelihood of being big D, little d. And then we multiply it out, q squared is 0.3 squared, which is 0.09, or 9% likelihood of being little d, little d. Again, this is showing you the likelihood of the individual having each of these conditions. So they would have a 49% chance that they would have a child that would be big D, big D, a 42% chance that their child would be big D, little d, or a 9% chance that the child would be little d, little d. The big thing about Hardy-Weinberg is that it's used to determine carrier probabilities, or when we hear that 1 in 500 births is affected by this condition, that's using Hardy-Weinberg. Also, looking at families and the likelihood that they would be carriers and then that their child might have a condition, that's using Hardy-Weinberg. So it's used for autosomal recessive diseases, where the homozygous recessive class is used to determine the frequency of the allele in the population, and its phenotype indicates its genotype. One such example is cystic fibrosis. The incidence in the case is you have 1 in 2,000 births. So what they've done here is this right here is the, let me get that there. This right here is the recessive. This is 1 in 2,000 individuals is little c for cystic fibrosis. So we take the 1 divided by 2,000, and that's where we get this number right here. So that's my q squared. To get q, I'm going to take the square root of that. Now, to get p, I know that p plus q equals 1. So p equals 1 minus q. So there's my p. To get the chance of carriers, I'm going to take my 2 times p times q, and I get this right here, which is 1 in 23 individuals would be carriers for cystic fibrosis. And so here you have carrier frequencies that we can do based on population groups, and you can see. And it's using the Hardy-Weinberg that allows us to make these type of calculations. So what is the probability that 2 unrelated Caucasians will have an affected child? The probability of both being carriers, we have this right here, the probability that their child will have cystic fibrosis. Therefore, the probability of 2 independent unrelated Caucasians having a cystic fibrosis child would be a 1 in 2,100 roughly, which is what we'd said earlier. It's about 1 in 2,000. We can use Hardy-Weinberg for calculating the risk of X-linked traits. For females, the standard Hardy-Weinberg equation is p squared plus 2pq plus q squared equals 1. But males, because males only have that one X chromosome, it's p plus q equals 1. So that if we looked at hemophilia, we have a 1 in 10,000 male births. So we're going to take 1 divided by 10,000. We get our number. Now that right there is my q. The p is 1 minus q, which would be this right here. The carrier frequency for females, we have 2pq. So 2 times 0.99 times 0.0001 equals this, which is about 1 in 5,000 births. The affected females, q squared, so about 1 in 100 million individuals, girls, could have hemophilia. As a reminder, p squared plus 2pq plus q squared equals 1. What that means is that when we add up our percentages should equal 100%. So this equation right here is showing us all individuals have been calculated. So Hardy-Weinberg is good for doing calculations of carrier states for genetics. That's really where it's important to be used. It's rare to be used for protein encoding genes that affect phenotype. It can be applied to portions of the genome that do not affect phenotype, such as DNA segments. But for the most part, we will be using this as doing calculations of carriers.